Hi everyone, got a full house today. Wonder who's speaking. Welcome to the Brain and Creativity Institute. Today we have the last talk of the Big Data and Human Behavior speaker series for this year. If our proposal gets approved to continue this series next year, you'll hear from me sometime during the summer. As always, I would like to thank Antonio and Hannah for allowing us to use this beautiful auditorium for our series. So as all you guys probably know, it's not easy to do interdisciplinary work, and in a lot of cases, and if not in most cases, interdisciplinary work does not result in breakthroughs in science. There's a simple reason to that. It's not easy to master more than one discipline. Our speaker today has done that, and by crossing boundaries and combining network science with neuroscience, he has changed the way we think about brain function. After receiving an undergraduate degree in biochemistry, Olaf Sporns earned a PhD in neuroscience at Rockefeller University and then conducted postdoctoral work at the Neuroscience Institute in New York and San Diego. Currently, he's the Robert H. Schaefer Chair, a distinguished professor and a provost professor in the Department of Psychological and Brain Sciences at Indiana University in Bloomington. Sporns was awarded a John Simon Guggenheim Memorial Fellowship in 2011 and was elected Fellow of the American Association for the Advancement of Science in 2013. Without further ado, join me in welcoming Olaf Sporns.
Thank you. Thank you, Morteza, for the invitation. Thank you, Antonio, Hannah. Great to see you, Irv, and Larry, and other friends here. It's great to come back to USC. This is my 6th time altogether. First time was 20 years ago. Irv had just moved here. In fact, on the way to dinner, you got lost on the way. We had to pull over to a gas station and figure out where Figueroa was. Anyway, it's great to come back and be among so many, be among so many friends and colleagues. And I'll talk about my work here fairly generally about brain networks and network science approaches to the brain and where things are right now, at least a small slice of that. I'll give you a brief introduction. I'll try to breeze through this as fast as I can, brain networks and connectomics, then look at connectome patterns of connectivity we see across different species. There are many commonalities that are kind of very interesting, I think, from an integrative neuroscience perspective. And then focus on various aspects of hubs and modules and relationship between structure and function in the human connectome. So first, a few words about brain networks and network neuroscience and where this comes from. I don't need to belabor this point here. The brain is a complex network of neurons and interconnected brain regions. We can study this network at multiple scales, individual neurons and their synaptic interconnections, as well as macroscopically the network of regions and interregional projections. We can ascertain those, we can measure those connections with various empirical methods of imaging, track tracing, or tractography methods. There's also 2 different domains of connectivity, which I'll refer to quite a bit throughout the talk. One is structural and anatomical, physical connections, real connections between individual elements of the brain. And then on top of that, relationships that result from dynamic activity and typically expressed statistically as functional connectivity. The network science framework ultimately involves a decomposition of a complex system such as the brain into elements and interrelationships. It's really fundamentally about relational data and the representation of relational data as a network representation. I'll refer to the elements as nodes, the connections as edges throughout. There's a huge, as I'm sure you know, a huge amount of activity in this field involving all sorts of networks, including transportation or human mobility networks shown here on the left that come into play when we're studying migration patterns of humans and things like epidemics and epidemic outbreaks in the population. Lending relationships among financial institutions which underpin the stability of the financial system, and relationships among proteins in cells, which many people in systems biology think are fundamental for understanding how cells, how a cellular state or global state is generated. Fundamental opportunities exist for us as in capitalizing on network science approaches within neuroscience. We can take on board a common quantitative and theoretical framework that is in part quite well developed in other disciplines. It does apply, I believe, although not immediately apply to our concerns in neuroscience. There's a wide range of computational tools we can use to analyze, to represent, and to mine our data, and there's the hope that network science might even offer us some universal principles of organization that underlie not just the brain, not just the economy, not just the social network, but all these systems together. There are many things that we need to be mindful of when we apply network science approaches in any given discipline, and especially in the brain. It's a fundamentally a very abstract representation about the system. Breaking a system down into nodes and edges that are fundamentally equivalent to each other does not pay much attention to the heterogeneity of the nervous system. We do need to take that on board more in the future, I think. We also have to pay attention to the spatial embedding of the brain. I'll refer to that later a little bit more in detail. And we have to get away from networks as being static representations of what's connected to what, and understand networks as dynamic entities that change across time and that have dynamics unfolding within them. Brain network data is hard to come by. Here we diverge from our colleagues in social networks, for example, where it's actually quite easy to acquire large numbers of, quote, big data from Facebook, Twitter, and you name it. It's actually very hard to configure good, robust, accurate data sets from empirical neuroscience data sets. We in the lab work with data sets coming from all the species shown here above, from C. elegans to the Drosophila nervous system, the rodent, the macaque, and ultimately the human. The fundamental pipeline, if you wish, or pipelines to construct networks from such data set goes a little bit like this. First, we need a decomposition of the brain into units. These can be individual neurons, as in the case of C. elegans, or they can be brain regions or local processing units or cortical areas in the case of higher species. That's a postulation in the case of the human brain, breaking down the brain into gray matter regions that are considered to be our individual elements. Then we measure with techniques that range from optical imaging to track tracing to spectrography and diffusion imaging, associations between these regions. We estimate how strongly they are connected structurally. I'm staying on the left side of this diagram for the moment. What is the, does a connection, a physical connection exist, and if so, how strong is it, and if so, in what direction does it go? This leaves us ultimately with a matrix representation, ultimately a big table of what's connected to what. All of the binary observations are aggregated into a complete picture, which is that connection matrix you see on the lower left, and that is what my colleagues and I decided some years ago, because of its fundamental importance for brain function, to call a connectome, a full set of connections of a nervous system at a given scale. That is the construction of structural networks. There are many methodological steps on the way here that are quite difficult to master, quite difficult to do well, quite difficult to do reproducibly. And that is a big challenge for us in the field at present, to do this well, especially in the human brain. On the right of this diagram is a different pipeline, a different processing algorithm, if you wish, starting with recordings of neural activity, perhaps from EEG, MEG recordings, perhaps from fMRI, perhaps from cellular recordings. That gives us time series of neuronal activity that can be processed once again into a set of bivariate estimates of statistical association between events in remote parts of the brain. This can be as simple as a cross correlation, often is in the case of functional neuroimaging, but it can be also much more complex in other contexts. Again, all these bivariate estimates of how two nodes in the brain are related to each other are aggregated together into a matrix form, which can also be rendered into a network or a graph. We call that a functional network, and we are going to be talking later in the talk about the relationship between the network you see on the left, the structural network, and the network you see on the right, which is a functional network and a cross-correlation matrix. Network methods, there's many of them. I'm only scratching the surface of the surface here in this little slide. There's an abundant tool set that we can use to dig into representational, relational data sets that come from the brain. Some as simple as, and as insightful actually, as node degree. The number of connections per node is very predictive of the influence that a node has, for example, in a social network or in economic setting. And it's also true, I think, in the brain. There's other measures that have to do with clustering, the decomposition of a network into motifs, and estimating the lengths of paths, communication paths, we think are very important in the brain because signals must travel through physical connections. Physical connections are not all-to-all. Very few neurons are directly connected to each other, only about 1 in 10 million, we think, in the human brain. So if messages or if signals, perturbations are passing through the network, they rarely do so directly from A to B, but there have to be intermediate stages in between. The length of these intermediate pathways are extremely important for the degree to which two elements of our brain can interact with each other. So looking at path length in a topological setting is actually a very fundamental thing that we look at a lot. Another very fundamental thing is decomposing a network into clusters, which are more densely connected within and less densely connected between. And it turns out that many brain networks can be decomposed into communities of highly connected nodes that quite often are found to be functionally associated with each other also in the context of physiology and behavior and cognition. Very important in that context then are nodes that are interlinking these clusters to each other, and these are the so-called hub nodes. I'm showing one of them here in this diagram. You can see right away why this node is crucial. If it is impaired, if it is perturbed or deleted from the network, communication between these two communities is entirely abolished, and that is obviously an outcome that could potentially have very severe ramifications. We're looking for hub nodes, and I come back to hub nodes several times throughout this talk because they are privileged, they are important, they are points of opportunity for communication, but also points of vulnerability in disease processes. Just a couple of wrap-ups here. So once again, I like to fundamentally and clearly distinguish between a structural network on one side, which is physical, which is real, which can be measured in different ways, where ideally the different ways should converge onto a single description. It has a finite set of elements. That's the notion of the connectome. It is a set of elements and connections that is finite and describable, and may have multi-scale organization as well. And then there's functional connectivity, a very different animal, if you wish. Statistical in nature, dynamic in nature, can be estimated with a great number of different measures, from cost correlation, as simple as that, to things that are more complex, information theory methods, Granger causality, you name it. There's a large, perhaps virtually infinite number of configurations of functional connectivity, even in a given brain, because the relationships in which brain regions are talking to each other, are becoming coactive over time, there's a very large degree of freedom there. It transcends what's underneath in terms of the more rigid anatomical skeleton. And I think a methodological convergence onto a single so-called functional connectome, I know people like the term, I actually don't like it very much, is quite uncertain for the reasons of this heterogeneity and temporal and time dependence. So in my view of this, and I have something at stake here, structural connectivity is the connectome, and then functional connectivity is what happens on top of the connectome, and if we want to, we can call it all connectomics. All right, so much for that. Going across species, the origin of mapping connectivity at the microscale in a whole organism go back over 30 years now to this network of C. elegans, which was published in the mid-1980s. A gargantuan amount of work, largely carried out by hand using lots of EM images of cross-sections of a few individual C. elegans organisms, reconstruction of a full synaptic network, chemical synapses, I believe shown here in red, gap junctions shown in a bluish tone. Maybe it's the other way around, I can't quite see it from here. That's a description that has largely remained stable for 30 years. It's the only organism for which we have a complete description of its synaptic connectivity, around 300 neurons and a few thousand connections. And the network is still being refined, remapped in some cases for related species, etc. But it's fundamentally, this is what it looks like, okay? What we're finding here, and this has been looked at over the years multiple times with different methodologies as the methods matured, we're finding that there are some neurons which are more highly connected than others. There's a so-called broad degree distribution. Some neurons have high connections Many of them don't. We're finding modules, we're finding communities of neurons that are more densely interconnected and are less densely connected between each other. We're finding some neurons which have, which have high centrality, which means they lie on many potential communication paths within the network. We're finding hubs. And we're also finding actually so-called rich club organization, which is that the highly connected nodes in this network tend also to be highly connected to each other, more so than expected by chance. I'll come back to the rich club feature a little later in the talk because it's actually something we've been looking at a lot recently. So even this very simple single cell scale network that we've known about for 30 years has a number of properties that are non-random, that are not found in a random architecture. We're beginning to get more data now in recent years, and in coming years we will get a whole deluge of big data, I guarantee you, on single cell connectivity patterns in portions of, and perhaps ultimately a complete mammalian nervous system, especially in the mouse. This is a paper coming to us from the Helmstetter group 2 years ago. This is in the retina, about 1,000 neurons in a small block of tissue, about 120 micrometers on each side, that were reconstructed, and synaptic contacts, which are not the same thing as synapses, were inferred from areas of proximity in neurites within this block of tissue. This gave these investigators a so-called contact matrix. This was part of their supplementary information. We've looked over the last couple of years at this matrix somewhat more closely to see if there's structure in this matrix and how these connections play out with respect to modules, for example. And one thing that we do find using a technique called stochastic block modeling is that there are clusters of more highly connected cells, interesting relationships to what cell types these actually constitute, and patterns of interconnections among them. This is a very highly non-random picture, and it's starting to reveal to us something about the organization of the mouse retina that perhaps we didn't know before. We will be, I'm going to predict here, next 5-10 years, we will see larger blocks of mouse brain being analyzed at the electron microscopic level, ultimately leading to reconstructions that will end up in graphs, in networks. Networks are a wonderful way of reducing what at the beginning are petabyte scale levels amount of data into a much more compact representation that can be statistically and mathematically analyzed. Going to another invertebrate, to another invertebrate brain here, Drosophila. We've been working with Enxin Chiang's group in Taiwan and Ralph Greenspan at UCSD over the past years to take the aggregated data that they get from individual imaging of neurons in the Drosophila brain mapped onto local processing units, so-called LPUs, creating a connection matrix from that is weighted and directed, and then looking at things like modules, rich club organization, etc. And there's a paper in press which will come out, we think, next month in Current Biology. We're finding, again, modules do exist, highly connected hub regions, and there is also evidence of rich club organization. Again, I'll say more about this later on. In the mouse, there's been a lot of activity at the mesoscale. My own efforts going back a few years, working with Andreas Burkhalter at WashU, looking just at 10 visual regions and their projections to each other, as well as to a set of targets outside of the visual system. And we found evidence for a sort of bimodal partition of this network into two processing streams that Andreas thinks are, based on physiological and anatomical criteria, are homologous to the dorsal ventral distinction that we find in the primate visual system. This matrix here is familiar to Hong Wei because he built it in his own work, coming from his data set, connected here of the mouse brain, a connection matrix published last year in Cell. And here's another matrix coming from an effort by the Allen Institute of the entire, this is, I think, a more extensive set of brain regions. And I know for sure that both of these matrices are being extensively looked at also from the point of view of graph theory and network analysis. But I want to highlight an effort that was started here by Larry and Mikhail Bota who, through hand curation of hundreds, if not thousands, of papers on track tracing data in the rat nervous system, built a representation of the interconnections of the rat neocortex that you see here, where each of the entries in this matrix is the result of not just one in many cases, but several observations from the track tracing literature of the existence of or non-existence of a pathway between two parts of the rat brain. Distinctive features here, this is a directed matrix that is very highly filled. 82%, I believe, of all the entries in this matrix have actually data that support either the existence or non-existence and the strength of a connection. This was due to really a gargantuan effort on the part of Mikhail putting this, collecting this data from all these different publications. There are observations we can make, global observations about how this network is mapped out. Once again, we're finding evidence for high clustering, short path length. These are sort of so-called small world attributes. We're finding modules. These modules, I was very pleased, did make some sense to anatomists who know a lot more about the rat brain than I do. We are arriving at these modules, mind you, not by using our knowledge about what we know about the rat brain, but using only connectivity information, only the graph, only the existence or absence of connections and their strength enter into the modularity analysis. And it's very data-driven that way, and it's pleasing to see that those decompositions of a brain network that you see here actually do make sense in light of what we know about anatomy more deeply and about function. And there's a paper coming out next Tuesday, so keep your eyes peeled. Next Tuesday, this will hit the newsstands, if not, well, the newsstands maybe not, but the anyway, we also find evidence for rich club organization. I'll mention this again. The fact that there's highly connected nodes, not only are they highly connected, but they're also highly connected to each other. I'll keep coming back to this and explain more about it a little bit later. Finally, the macaque, of course, had been looked at for a long time with efforts there and compiling databases going back all the way to the early 1990s. Some seminal work by Dan Fellerman, David Van Essen back then, which really got me and others started in the field of looking at graphs, because what they published here as a table really was an adjacency matrix and a mathematical object. The efforts carried forward these days by more recent analyses of macaque connectivity coming, for example, from Henry Kennedy's lab that are painting a somewhat different picture of what you see on the left. On the left, much sparser binary representation fundamentally among connections being either present or absent. On the right, a weighted distribution of connection strengths and a lot of pathways that are actually quite weak. And we're still all processing what this means to the architectural properties of networks that we find. There are a lot of common properties here. I want to underline this. We're finding in all these different species, going all the way back to C. elegans, we're finding that there's a broad degree distribution. The alternative hypothesis would be all neurons, all brain regions are equally well connected, have the same number of connections by and large on average. Instead, what we find is there's a broad distribution. I don't want to call it scale-free. It has a heavy tail. There is a small admixture of highly connected regions or neurons, and we find this across different species. It's a finding we also come across in the human brain using diffusion imaging and tractography. I'm very pleased we see it also in animal data sets that are not acquired using those more indirect inferential technologies. We're finding that each region, this is true for the rat brain, for the mouse, for the macaque, it's true for C. elegans even, every node has its own very specific set of inputs and outputs that we and many others believe plays an important role in defining what it does. In our network, fundamentally, who you are, quote unquote, what you do, what your specialization is, depends on who you're connected to, what your inputs and outputs are. It's not so much your intrinsic capacity, but it's who you're talking to that defines your functionality. High clustering, short path length, interconnected modules, and a tendency to form a core or rich club of highly connected nodes that are also highly connected amongst each other. Those are common themes Now, you might say, so that you could take a negative view of this and say, so you really haven't found anything interesting yet, right? Because if every nervous system you're looking at has these properties, well, so what if you told us that we didn't know already? Well, first, we didn't know this a few years ago, but I would put this the other way around and say maybe some of these properties have reflect some fundamental some fundamental requirements on functionality that can't be contravened by any nervous system. Maybe there's a need, I'm speculating now, for something like a rich club organization to exist. Maybe there's a fundamental requirement for all nervous systems to have an anatomical substrate where information can come together, okay, and can be shared. Maybe this is true for a humble worm as much as it is true for a proud primate. We're looking at trying to explain these features of brain networks through the use of so-called generative models. Generative models are very simple very simple connectivity rules or wiring rules that can be instantiated in a computer program that are driven by very few rules and parameters and can generate connection matrices. You can think of them as an encoding. If you think of a given empirical network, you can ask, what are the rules, the minimal set of rules that I need to generate this network from scratch? Those rules could be things like a distance rule. If two nodes are connected, if two nodes are closer together in space, they have a higher probability of being connected. That's a simple rule I've just given to you in a non-mathematical way that you can write down as a mathematical statement and put a parameter in there, which is your decay with distance, and you can build networks based on that rule, and you can compare them to what you actually see in reality. We've been looking at this quite closely and have a paper in, well, I was going to say in review, but we're not quite sure yet, that is getting at that, and the upshot of that paper is, I think, quite interesting because space, and we know this since the days of Cajal, is the economy, the drive towards economy, towards minimizing material and space and other resources like that. That's a driving factor in how neurons and circuits shape is dictated. But it's not the only story. We find in all brains we've looked at features that violate the minimization constraint, that essentially are instances where the brain is doing things it shouldn't be doing if all it was doing was minimizing wiring. We are very interested in those particular features. Our thinking is that it is those features of brain networks that tell us something more about function, about a benefit, about something that has been, about some value added that is coming out of this arrangement of connections. If there wasn't any value added, this would have gotten rid of, because the wiring minimization constraint is fundamental and cannot be overridden for many reasons. So, long story short, and in fact, hopefully there's a paper eventually, if the reviewers allow us and the editors to get this published. There's a trade-off here. We can't explain all features of, let's say, human brain networks through wiring minimization constraints only. We can explain some additional features through topological principles that ultimately have to do with the existence of rich club organization and the information sharing requirement across different domains and different modules between brains. And those generative models do a very good job of regenerating human brain networks, for example. But that's a story that's still in progress. I was hoping I could tell you more about this here, but like I said, we're still on the road with that. A couple of words now about the human connectome and hubs in it. This was an early draft of the connectome, 2008, work done with Patrick Hagman, who developed at the time the sort of quite ingenious way of building these networks from structural imaging, gray matter separation, dividing the gray matter up into free surfer regions and then subdividing those free surfer regions further into geometrically about equally shaped so-called regions of interest, a tiling of the cortical surface, in addition subcortical regions which are not shown here. Then on top of that, doing a very, I think still considered to be very high, relatively high fidelity, high resolution approach to diffusion imaging and tractography called DSI, diffusion spectrum imaging, which handles areas of complex fiber architecture with greater accuracy than DTI does. From that, we're getting what I call a colorful hairball that you see there, streamlines that have colors that mean nothing. So those are our edges. We have our nodes on the left. These come from gray matter parcellation. We're pulling together, this was our idea back then, which now everybody is doing. Nodes and edges, we're putting this together, and now we're getting a network from that. We looked at this with all the tools we had at our disposal at the time. We have done this many times over now. This has been replicated many times now in other imaging facilities. Diffusion imaging photography of the human brain is carried out everywhere now. We're finding unique regional fingerprints of connections. Again, every region, every little tile of the cortical surface has its own set of inputs and outputs. We think it's very informative about what it ultimately does. There's a broad exponential, although not scale-free, degree distribution. That is, we have a small mixture of high-degree hubs. There's high clustering and short path length. We find modules interlinked by hubs, and there's a structural core. At the time, we called it a structural core, not a rich club yet, but it's fundamentally a related idea. With a prominence, actually, in a part of the brain that I know Antonio is deeply interested in. The second to last time I was here, we talked a lot about this. The posterior medial cortex, the precuneus posterior cingulate cortex, a part of the brain that is not only highly connected, has many connections attached to it. We know this also from, for example, Antonio's track tracing study in macaque, but also those bits of brain that sit back there are very highly connected amongst each other as well. And so if you do a so-called K-core decomposition, it's the part of the brain that's most resilient to erosion. It is actually very highly connected and highly connected within itself. I'll come back to that idea of the core and the rich club in just a second. Many studies have been carried out to map hubs and structural connectivity in the human brain since then. While there are many methodological divergences between different acquisitions, different ways of doing even just the physics of the acquisition itself, the post-processing, the tractography. There's, I think, a pleasing degree to which the observations come into some overlap with each other. I'm showing a few summary diagrams here from our own early study, the study by Gaolang Gong, upper right, and then more recent analysis by Emil Nijhuis at the bottom. And for instance, the prominence in structural connectivity analyses of the posterior medial cortex does hold across different acquisitions, across different imaging centers. It does tend to pop up as a highly connected, very densely connected part of the brain in other studies as well. So that's encouraging from the point of view of there's something going on here that's of interest, perhaps of deeper interest. Detecting hubs and functional connectivity is somewhat problematic, although I keep getting papers to review where people use things like degree. They should not. This is not the way to do it for reasons I can't get into. In fact, Jonathan Power wrote a paper about this a couple of years ago. I think it came out in Neuron in the end. And one of the better ways of diagnosing hubs and functional connectivity is to look at so-called participation coefficient, which is the degree to which a given region participates in its functional connectivity with not only its home community, which is its own backyard of other regions to which it's strongly connected, but also others. And that is, for methodological reasons that I can't get into, a robust and sensitive way of detecting highly connected hub regions in functional connectivity. Degree and betweenness centrality, for example, do not make sense in that context. Hubs are popping up in literature over and over again now, and in part for reasons because they are implicated more so than other parts of the brain in the pathology, in pathological changes underlying brain and mental disorders. The table that you see at the bottom here comes from a recent meta-analysis by Nick Crossley, in which he looked at structural scans acquired from patients who were diagnosed with a broad range, I think about 30 or so brain disorders. You see them all listed on the left. Among them, some very common ones like autism, schizophrenia, Alzheimer's, etc. And he asked the question, in those structural scans in these patients who have these brain disorders, where do we see impairment? And he looked essentially used a normative connectome map to say, well, those regions where the impairment starts, do they have high degree or low degree? So he took a connectome map and said, in these impaired regions, what is the degree to which these regions are central, to the degree to which they are hubs. And it turns out that there's a tendency across many or most of these conditions for them to start in highly connected parts of the brain. This is true in spades. We think now, when people think in the field, in Alzheimer's disease, for example, which starts in key hubs in the medial temporal lobe and then spreads to other key hubs along the midline. There are other conditions where it looks as if hub regions, impairment of hub regions is one of the triggering events. If for no other reason than for these clinical implications, hub regions do pop up in analyses and are therefore important to take into account. Now I'll get more into the Rich Club. What is that exactly? On the left, in this little diagram, you're seeing some communities, a schematic diagram of some communities and how they're interconnected through highly connected hubs. And perhaps you can see right away these hubs are crucial for inter-module communication, but they're not really sharing information very efficiently amongst each other, these hubs. In the middle diagram, there's additional connections that link hubs to each other. And if we looked at the statistics of this, we would find that if we just look at the number of connections we find on each node, the nodes in red are more densely interconnected amongst each other than we would expect if connections were falling into place just at random. It's as if there was a drive to create these connections or to maintain these connections in the first place. And that's the essence of our so-called Rich Club organization. It comes from social sciences, and the connotation there is that, highly influential, highly connected individuals in social systems quite often tend to be connected to each other. Why is that? Well, they're the 1%ers, right? They are the ones that are giving themselves to low tax rates. Influence, right? It's influence. It's about information sharing and influence in social systems. Here it's probably something different, but we do find this type of organization in the brain. It's not, no pun intended, a no-brainer that we find it, because there are biological systems where a Rich Club organization is conspicuously absent. For example, protein networks show the opposite mode of organization. Highly connected proteins which bind to many other partners, tend to not bind to each other. Perhaps one could speculate as to why that might be. Starting in this line of investigation a few years ago with Martin Mannenheuvel, we set out to look for Rich Club organization in an initially small cohort of participants using both low and high resolution mapping. And the key curve here is the red one. The axis, the peak, shows you that there's denser connectivity at high levels of K among high degree nodes. Then compared to our random model where the degree is preserved but the connection pattern has been randomized. We find more connections among the high degree nodes than we expect to see based on a simple, perhaps liberal, but still, I think, admissible null model. Where are these Rich Club nodes and their connections? You see an anatomical diagram down below. They are scattered throughout the brain. They're including regions like the posterior cingulate cortex and precuneus, portions of superior frontal cortex, medial orbitofrontal cortex, all of which, by the way, are subcomponents of the default mode network. I'll say a bit more about this in a moment. Some other cingulate regions, the insula in both hemispheres, and the medial temporal cortex. An interesting thing that we looked at was if this is our network, if this is our structural network, and we assume for the moment that it is correct. Mind you, diffusion imaging cartography has many problems associated with it, so I say this with a grain of salt. It is, however, the best picture we had within this study at the time. It's the best reconstruction we could come up with. If we assume that we are now wanting to communicate between all parts of the brain, all pairs of nodes want to, let's say, send a message to each other, and if we assume that that message is best relayed on the shortest path, which makes sense, it's the path least corrupted by noise or by intermediate processing steps, least attenuated because you have the fewest number of steps. So the shortest path is somewhat privileged. Let's assume we're taking the shortest path from any point to any other point. How often do we pass through the ridge club? It turns out that we're touching a ridge club node almost 90% of the time, and we're traveling through two ridge club nodes and a connection between two ridge club regions 2/3 of the time, despite the fact that the ridge club only takes up about 10% or 12% of the total infrastructure of the brain. So it's a part of the brain that's about 12%, and it soaks up message traffic, hypothetically speaking. Mind you, this is just a computational exercise. We can't actually follow messages around in the brain at this point. We can't do that. But in a modeling setting, it soaks up traffic quite a bit more than what it's actually constituting. This is an analogy here to the highway system, which in the US makes up about 40,000 miles. This is about 1% or 2% of the total road system of the United States, but about 25% of all vehicle miles are unfolding on the highways. And we think for reasons that have to do with highways are efficient ways of communicating, especially or transporting vehicles and people over long distances. And there's an analogy here. Perhaps we shouldn't take it too far, but in our thinking, that's how we think about it. Okay. Relationship of ridge club and resting state networks was an interesting next question for us to explore. Resting state networks are fairly consistent decomposition of the brain into functional modules or functional communities. We can achieve that decomposition through network analysis, or we can achieve it through ICA. Those give fairly equivalent answers in the end. And we know that resting state networks have an underlying anatomical basis. The components of them, which are often quite remote from each other, are connected through white matter pathways. We also know that there are non-stationary fluctuations in functional connectivity. Having said that, actually accurately, in a statistically valid way, extracting these is a non-trivial exercise and somewhat of an ongoing research project. But I think the data points unequivocally in the direction that there are at least some fluctuations in functional connectivity across time. And our question then was, well, we had just found the ridge club. We had also found it in macaque brain at the time. So how does this relate to the overlying functional networks that have been studied a lot in the context of cognition and behavior? Is the ridge club just one of them? Is it the default mode network, for instance? Or is the ridge club more broad than that? Does it interconnect multiple functional networks to each other? And working with Martin Mannenheuvel, we carried out the following, or he carried out, I should say, the following ambitious study. Gray matter pacillation followed by imposing a node pacillation on the cortex and the subcortex gave us our nodes. Diffusion imaging tectography gave us our structural edges, and functional resting state functional connectivity and ICA in this case gave us our functional modules. Now we could do all kinds of cool things like overlay the functional modules on top of the structural connectivity and look at things like the ridge club itself. How does it actually intersect with those functional modules on top? Here's a side-by-side of the structural connectivity on the left with diffusion imaging tectography and functional connectivity on the right with fMRI, resting state. Two different imaging modalities. I think you can see that there's a pleasing degree of relatedness between these two patterns. I say pleasing because they are two different imaging modalities, two different acquisitions. If they were totally unrelated to each other, one would have questions about either one or the other or both of them in terms of whether, well, there's still a possibility there's a common artifact, right, driving all this, but I try not to think about that. Anyway, there's couple of low-hanging fruit here that I briefly want to breeze through towards the end of my talk already. How do witch club nodes break out with respect to the overlying resting state networks? The majority, almost half of them, overlap with 3 resting state networks: the default mode network prominently, the salience network, and the frontoparietal control network. Those, all 3 of them jointly, you might call them in quotation marks the more cognitive resting state networks, the more multimodal networks that are engaged in cognitive control, self-related processing, high-level cognitive functions. Unimodal networks such as vision, auditory, somatosensory, motor are less participatory, participate less in the witch club, but all have some participation. So the witch club ultimately connects together all resting state networks with a strong emphasis on those resting state networks that we think are engaged in higher cognitive functions and in cognitive control. The model in the end looks sort of like this that we ended up with, as you see here down on the lower right. In pink in the middle would be those more higher order cognitive networks, default mode, saliency, frontoparietal, and then the satellites off to each side might be our unimodal sensory motor appendages, and that's the picture that we came out of. So the witch club, highly connected nodes, the highway system of the brain stretches out over all of its functional subdomains with a heavy representation, heavy anchoring, if you wish, in those more highly cognitive networks. Finally, a few words about relating structural and functional connectivity. This is a topic that is of very great significance to me. I'm ultimately a computational neuroscientist, and I like to build models that have certain physical specifications, certain parameters that we put in, that can generate neuronal time series that we can then compare to empirically recorded neuronal time series, and to tell us something about where these functional relationships that we think are so important in the brain come from. So I'm not interested so much in functional connectivity per se. I'm interested in where it's coming from. Underneath, what are the generative models, once again, that can generate those resting state networks that we see empirically and that we think are important for our cognitive architecture. So we know that functional connectivity has an anatomical basis, at least in part. Not totally, because remember, it's fluctuating a lot on a fairly stable anatomical substrate. So the fluctuations are not easily explained by what we see in the anatomical substrate itself. But there is a shaping effect of the underlying anatomy. We know this from a number of different studies, coming from macaque initially, the study by Justin Vincent some years ago, and then our own studies in the human brain. If we compare structural connectivity, functional connectivity side by side, there's a relationship between them, a statistical relationship. We also know that resting state networks are intrinsically interconnected, and we know that when we cut structural connections, as is sometimes the case in the case of, for example, callosotomy, we measure functional connectivity before and after, we're seeing dramatic effects of that perturbation play out in the human brain. So structural connections are important for generating and maintaining patterns of functional connectivity. My former graduate student, Chris Honey, now professor in the University of Toronto, did all these studies with me back in the day, looking at structural connectivity, trying to predict from structural connectivity what the functional connectivity pattern might look like. The much more difficult inverse mapping of inferring the existence of the presence of anatomical connections on the basis of neuronal time courses. This is a very messy inverse problem. I think it's ultimately not going to work that well, but it can at least be attempted. And then using structural connectivity to model explicitly built computational models of the brain in a computer that can generate time series that we can then compare to empirically recorded time series. That latter effort started out in our case with the binary matrix of macaque interconnectivity in the visual cortex coming from Van Essen's work largely in COCOMAC, using this matrix as our coupling matrix among nodes which were operating under the guidance of biophysical equations that described largely what these nodes were doing dynamically at each point in time. This combination of biophysical equations, which define node dynamics, and structural coupling, which is the connectome, gave us neuronal time series, which we passed through a hypothetical fMRI mechanism to give us fMRI, synthetic fMRI signals, which you can then process into a matrix of bold cross correlations. So this is doing MRI without a scanner. It's a much cheaper way of doing MRI. You don't have to pay your subjects. You can do whatever you want. You can do great things, like you can perturb this matrix. You can throw away connections or entire brain regions with their inputs and outputs, and you can ask, how does that affect the pattern of functional connectivity? You can make a prediction about that. We were at the time, 2007, we did not have macaque data to compare to. This is data that's quite precious to come by. There's not many labs in the world that can do resting state connectivity, resting state fMRI with macaque monkeys. They're very uncooperative, I'm told. Only years later, Yasushi Miyashita at the University of Tokyo contacted us wanting to get our model data to compare to 2 data sets that he had acquired in 2 monkeys that were actually anesthetized and put into a scanner, and then looking at spontaneous brain activity in that state. And here's the side-by-side of that. To our relief, I guess, our very simple, extremely simple biophysical model, just 3 equations per brain regions, a few parameters, and a binary matrix that was lacking 25% of the brain, actually, 1 hemisphere only. We were able to come up with a pattern that you see here on the right that is not completely off. It's not totally accurate either, but we think that with more accurate data sets as our input, and perhaps more accurate representations of the generating mechanism for the time courses, we could do a better job with this. So it's encouraging because it tells us something about, we can in fact build a generative model for functional connectivity based only on anatomy and biophysics. Okay, so that's the idea here. We took this over to the human domain. The empirical patterns here on the left and in the middle. On the right is a simulation. Glass half full, half empty. It's not totally accurate, but also not totally unrelated to the empirical pattern. More recently, we looked at communication processes in greater detail. We wanted to get away from the detailed biophysical simulations. One way to go is to make your simulations more accurate, to put in layers, to put in different cell types, to put in spiking neurons, which we didn't use, to build a more and more accurate representation of the human brain. As a theoretician, I wanted to go in the other direction, against the stream, against the funding stream, also, it turns out. But, I have tenure, I don't care anymore. So, I actually wanted to abstract away as much as possible from brain communication processes and see if we could still come up with a prediction that's reasonably accurate. So we went to the literature on transportation networks, navigation, how people navigate in cities, communication networks in computer science. And there's a big literature there that people have looked at over the years. What are the patterns of navigation, of communication in these networks, and how has the network topology been shaped to enable efficient navigation and communication in such networks? This could be a design process if it's a technological network. It could be an evolutionary process if it's a biological network. And so we thought of the brain, we proposed, let's think of the brain as a communication network. Let's just think for one moment that communication is of the essence, and that the topology of the brain has been organized to enable efficient communication, to enable different parts of the brain to, quote, get in touch in ways that are unfolding along short communication paths on average, and enable efficient signaling between them. And we built an analytic model for functional connectivity that runs not as a simulation but as a simple matrix algebra exercise, which is great because the simulations take about a day or 2 to finish on a single core, but these models run in under a second. You use MATLAB, you hit the return, and you get an answer back right away. I love that. Much better than waiting a day or 2. So here's a couple of things that I think we can all take for granted. Imagine you're communicating from source to target here, and I'm done in just a minute. Okay, this is my last. This is my last. My last rigmarole. I got a long introduction, so anyway. So let's say we're going from source to target, and let's consider situation A first. The situation on the left. The situation next to it, source to target, those connections are weaker, and it stands to reason that the communication from source to target is somewhat attenuated here. It's somewhat weaker, it's somewhat less, okay? And if we could measure functional connectivity, the degree to which time courses on source and target are aligned with each other, it stands to reason that the case in the middle is would result in less functional connectivity. I think there's not much argument. If we add another step to the chain, now we have a longer path. Again, it perhaps stands to reason that functional connectivity is again attenuated because signals there's more opportunity for noise, signal degradation. The signal it doesn't get stronger as it gets passed along. It tends to get a little weaker and attenuated. There's two other situations that we considered. Imagine that you're going from source to target, but it's not just a mindless, stumbling forward as in the middle, but there's off ramps. Let's say you don't quite know where you're going, and suddenly you're in Times Square. And now you have to make a decision, where's the hotel, or where's my destination? This is a navigation problem. People do that all the time. You need a lot of information now. From a statistical point of view, you can actually express it as a Shannon entropy. The more options you have, the more information you need to make the correct decision. And we can actually quantify the amount of information you need to find the shortest path from source to target as the sum of the Shannon entropies of what information needed each step. That's called search information. It's a measure that existed in literature, in the statistical physics literature, and it can be quite easily computed if you have an underlying connection matrix, for example. I'll leave aside the path transitivity for economy reasons now. And it turns out that we can predict functional connectivity with a weighted combination of connection strength, path length, search information, and path transitivity very nicely. This is a somewhat fancy representation. The left triangle of this diamond-shaped matrix are empirical recordings of fMRI signatures coming from the resting brain of a number of participants on 500 nodes in one hemisphere of the cerebral cortex. The right part of the triangle is a prediction based on an underlying connectome network, which I'm not showing here, and those four measures of communication that I showed you earlier: the strength of the connection, the length of the path, the search information needed, and the path transitivity, which I didn't talk about. And this is an analytic model. It's not a simulation. It's actually something that you can compute directly on the graph. And the accuracy of this prediction actually exceeds what we do with our lengthy neural mass simulation. It's better at predicting resting state functional connectivity than our somewhat messy and laborious neural mass simulations. On the right, there's a few seed maps of different regions in the brain that we think are differently well captured with diffusion imaging. That's why we're doing, in some cases, very well, like in the visual cortex, and in the lateral. In the temporal lobe, we're doing considerably worse because we're lacking all of the interhemispheric connections that tractography simply doesn't find in the human brain for reasons that are beside the point. So this is a communication model, analytic, makes a great prediction. We've been building on this now recently, going into integrated detail on this and digging more into it. Final point I want to make is another way of modeling communication, quite abstractly, is to use a dynamic model, yes, but a more abstract model than a neural mass model would be. And that's work done by my postdoc, Bratislav Misic, who at the time was a graduate student with Randy McIntosh. And that's using a message passing dynamic paradigm implemented on a macaque, in this case macaque brain connectivity data set. And I will go to the second slide right away and say that this dynamic model has helped us to identify, in our latest iteration of this, what we called convergence zones. That is, parts of the brain where message traffic aggregates and that are to some extent bottlenecks for communication in the brain, because as messages converge, they have a hard time getting through. One of the convergence zones that was flagged in this work was, for instance, the hippocampus, which does not show up prominently in many graph theoretic analyses as a hub, for instance, but shows up prominently when we put dynamics on top. And I've always been very fond, and Antonio knows this, of Antonio's work in 1989, published in volume 1 of Neural Computation. I was a graduate student back then, but I remember reading this paper, and everybody was interested in the binding problem back then. This was a big topic. And we have remained very fond of this notion of convergence zones ever since. And so we talked about this earlier in our conversation. I think there is a connection here between the kinds of ideas that you've developed over the years about convergence zones and what role they play in cognitive function and behavior, and the kinds of more, I would say, data-driven, model-driven analyses that we've been doing on connection matrices, where we find not only topologically highly connected nodes, which are prepositioned to be such convergence zones because of their connectivity. They're also connected amongst each other. This is the rich club. And then we find that when we implement dynamic processes, communication dynamics, there's actually additional nodes that pop out, such as the hippocampus, that are not captured well with graph theory. So we hope that we can continue that conversation about what convergence zones might be and what the relationship might be between these types of phenomena and the things that you're talking about in your work. That gets me to the end. Okay. Well, there it is. Network science is important. There are hub nodes, and they are interconnected amongst each other. We hope that, I hope that the interconnection between neuroscience and network science methodologies and modeling methods will continue to be fruitful for people who are interested in integrative properties of the human brain and other nervous systems. I'll say one thing more about it. Unlike other analysis tools like ICA or MBPA or what have you, networks are different. Networks are a theoretical framework. They are not just a way of getting a number from your data set. They're not just a way of getting a t-test so that you can write a paper about it. They are actually there's a theoretical framework that has deep roots in mathematics and statistics that not only tells us, like ICA does, what are the components in a given architecture, but it also tells us about how they're connected. We can apply this framework iteratively. We can build generative models that explain network topologies. We can build dynamic models on top of networks. So it's a framework that really is extremely rich, and I think has the potential of also unifying different efforts in neuroscience more so than has been the case in the past. I'm beginning to talk more and more again to people who are doing single-cell mapping in invertebrates and in the rodent brain, where ultimately these network modeling methods will become extremely important for reducing the massive big data. I've mentioned it at least once now. The massive big data that come our way can be mapped into a low-dimensional space that we can actually understand and work with. So I think that networks as a unifying framework, structure, function, and different scales of analysis have a long way to go. Thank you very much for listening to this.
In the functional networks, the starting point is generally computing these five area correlations or other interaction measures, which is going to give you a link in a graph whether there's a direct interaction or indirect one. So I wondered if you looked at, say, computing partial correlations or other multivariate graph
yes.
points, whether your hub interpretations are the same or when you find...
Yeah, good question. So it embarrassingly perhaps, simple correlations, full correlations are the measures that still make to us the most biological sense. We've looked at partial correlations. I know that Steve Smith is very in favor of them, and we've computed them and we looked at them, and they make no sense to us whatsoever. We cannot We cannot build generative models, for instance, coming from this underlying structural connectivity. So despite the fact that full correlation is relatively simple and is a correlation, not a causation, we all know this, a simple statement about the similarity of time courses, which is a statement about how differentially two brain regions or two neurons might respond to a variety of inputs. That simple statement is still remarkably more informative than a much more sophisticated but also somewhat brittle decomposition into full partial correlations where you partial out everything for each pair. This doesn't have a biological meaning to me. Okay, I don't know what that means. I know what a physical connection is, and I know what a correlation is, similarity of time courses. And if one is careful about interpreting both, one is fine. I think there's other ways of looking at bivariate relationships that might be in the future as we have more data. Might be useful, like mutual information would be one of them, because we're capturing nonlinear relationships in addition to linear. I used to work a lot with transfer entropy, which is a information theoretic variant of Granger causality that tells you something about directionality for various reasons. Not great to use in fMRI context, but great to use in a single cell context. We've got a paper in the works with our colleague John Beggs at IU looking at information flow in highly resolved neuronal circuits, where we basically also find rich club organization incidentally. Highly interactive neurons are also highly interacting with each other, more so than expected by chance. Yet again, another example of how this theme carries through. So I actually think that full correlation have a way to go here, and despite the fact that they're simple and perhaps too simple, at least it's something that we can interpret.
I was just wondering, you mentioned big data twice. What aspects of advances in big data analytics would benefit brain networks the most in your mind?
So almost by definition, if you're doing imaging these days, you are in the big data arena, because there is more and more emphasis now on large collections of usually public repositories of at least at the primary level, very large data sets. Once they are reduced to matrix form, for instance, they're actually not, in my opinion, big data anymore. They're actually already quite manageable. But we were talking about this morning. I mean, I feel conflicted about big data, in part because I'll repeat what I said in the car. There's no future that I see going forward where the data isn't going to get bigger and bigger, right? It's never going to get smaller again. It's going to just get bigger and bigger. But as that happens, there's also an increasing need to reduce these data to small data, because ultimately to do science and to have understanding, we want small data. I'll give you an example. Hubble, right, who discovered that the universe expands. That's a major discovery, PNAS paper, right, 1929, I think, based on a single scatter plot, essentially, of the redshift of I think 44 galaxies or something, and a distance or something like that. I've forgotten the exact axes now. That data set fits handily on a flash drive. It's a couple of kilobytes, and it had an enormous impact on our understanding of the universe. It's coming from big data, if you wish, because going into that data set were many spectra that he took from astronomical observations, images of galaxies in various forms that have probably many megabytes, if not gigabytes of data combined as a combined total. But the data was reduced to that scatter plot through applying theory, knowing something about how things should behave, having physics at your disposal, which unfortunately we don't have in the brain yet. I think the challenge for us in neuroscience is to come up with those guiding principles, if you want to call them laws perhaps, that allow us to reduce these enormous data sets that come our way, to identify the low-dimensional subspaces that we should be mapping these data into, that we can then interpret. Unfortunately, that process is underappreciated, I think. If you want to get funded, please don't say that. They might be recorded. Yeah. Anyway, because it's all about getting more and more data these days, and that's great. And I'm not saying it's bad to have more data, but I think we have to have there's a real need for identifying how do we map these huge amounts of observations into what are the relevant dimensions, right? And there I think we're just at the beginning, certainly in our field. I tend to hope that networks graph theory are one way of doing this. They're one way of decomposing a system, one way of abstracting away some things and focusing on others. This has to be done carefully and with understanding of what one is doing, but I think it's one way of doing it.
Thank you.